Okay, so hi everyone. My name is Santiago. I am a PhD student working with Ana Amador at the Dynamical Systems Laboratory. So I wanted to begin by thanking the iPoLS organizing for inviting us. And I'm going to be telling you a little bit about the work done at the lab. First, I'm going to give a broad context of the work done previously before I entered my PhD. And then I'm going to tell you about the experimental work that I have done at the lab. So in order to begin, the first question is always why study songbirds? And the thing is, if one wants to study vocal production, the vocal production mechanisms in songbirds share many similarities to humans, mainly because the songbird's vocal organ, which is called the syrinx, works in a similar way to the human larynx. That is, these are membrane tissues that oscillate and then they modulate the airflow coming through the lungs or the air sacs, and that perturbation in pressure travels throughout the vocal tract and eventually comes out, and it's the sound that we hear. And not only that, but songbirds, which are about 40% of bird species, also have to learn to vocalize. Their vocalizations are not innate. They have to learn by tutor imitation, much in a similar way in what we humans do to learn language. And this is a rare feature in the animal kingdom that we share with elephants, cetaceans, bats. And so if we want to study this at the lab and conduct experiments, songbirds like zebra finches are an easy animal to manipulate at the lab. So songbird production, bird song production can be explained in a simple manner by saying that there are motor commands coming downstream from the brain to control the muscles that surround the syrinx, which is the vocal organ. And as I said before, this generates a pressure perturbation at the sound source level that travels throughout the vocal tract, and that is eventually the sound that is produced. So here we have a typical zebra finch song. The upper figure is the sound trace, and the bottom figure is the sound spectrogram. And so I'm going to play back here. So that's the sound. It's not a very glamorous song, but the thing with zebra finches is that they learn their song when they are young, and they repeat the same song throughout their lifespan. So each individual of this species learns a given sequence of these units that we call syllables, and eventually what they consolidate in their memory is a song motif that they repeat throughout their lifespan. And so if we want to study this, the two main questions that we can ask is, how is this produced at the vocal organ, which is to study the peripheral biomechanics, and how are the motor commands encoded in the brain? And so there has been a lot of work done at the lab by Gabriel Mindlin and his students throughout the years. This is about 10 years of working to model the working principles of the syrinx. So what we have in this picture is one of the sound sources that form the syrinx, which are membrane of connective tissue that can oscillate. And they have modeled it as a nonlinear oscillator. Here are all the terms of the equations. I'm not going to get into very much of detail here, but you can ask me later. And what's important about this is that we have some terms of nonlinear restitution and nonlinear dissipation. But more importantly, we have only three parameters, one of which is, two are time dependent. The first one is alpha and is related to the air sac pressure in the sound source. Another one is related to labial tension. And the third is a static scaling parameter. And so with this modeling, we are capable of producing synthetic sounds by varying the alpha and beta time trajectories in parameter space And we can generate a lot of different complex sounds by different mechanisms. This system has two bifurcations, which are a tool that is inherent to study nonlinear dynamics. So this system has a Hopf bifurcation in which oscillations are born with a defined frequency and zero amplitude, and the saddle node in a limit cycle in which frequencies are born, oscillations are born with defined frequency and zero amplitude. So they generate basically tonal sounds for the Hopf bifurcation and spectrally rich sounds for the saddle node in a limit cycle. And what's important is that now at the lab we can reconstruct alpha and beta parameters directly from a song recording and produce a synthetic song. And more importantly is that these two free parameters of this low-dimensional model are related to physiological variables that the bird can control during singing. So it allows us to interpret song production in terms of motor coordinates that the bird controls. So over here we have both the sound trace and the spectrogram for a recorded song and for a synthetic song. And so I'm going to play back to you this. That's the recorded one. So again. So this is done by numerically integrating the differential equations that I showed you before, the model. And so what we have here is a nice result. And we can see that it sounds like a Seraphin song. I hope to have convinced you about it. And that it looks like a Seraphin song. I mean, the acoustic properties that are shown in the spectrograms are similar when comparing both cases. But the question remains of whether the songs generated with the model are biologically relevant or not. In the sense that I'm going to tell you right now. So here is a zebra finch brain, and what's important here, I want to remark the blue pathway right here. So at the top we have HVC, which is the high vocal center. HVC is a sensory motor nucleus in the songbird's brain. This means that it integrates both auditory information and is involved in the production of the vocal behavior. And so information comes downstream from HVC and eventually reaches these three nuclei. One is N12, and then there are RAM and PAM. These two control expiration and inspiration, and this one, they directly innervates the muscles that surround the syrinx. So what's important about this is that neurons in HVC respond selectively to the bird's own song. This means that neurons at the HVC level are involved in producing the song. I mean, it's encoding some features that we yet don't know because it is an open question in the field. But also, remarkably, when you play back a recording of their song to the birds, neurons in HVC will fire, and they will fire in the same way that they fire when the bird is singing. So this is great for us because it serves as a basis to test the biological relevance of our model, in the sense that we can replay the automatically reconstructed synthetic song, and then see if these neurons in HVC that are finely tuned to the bird's own song fire or not. How am I doing? So this is right now is the work that I started doing at the lab. I began working for my master thesis, and then I stayed for my PhD. So without entering too much detail about this, what we are going to be doing is measuring neural activity at the HVC level. And for that, we need a stereotactic frame in order to look for the coordinates of HVC and lower an electrode. And the thing is, commercially available stereotactic devices usually use a system of barring the ears to hold the head fixed. And that is no good for us because we are going to be dealing with auditory stimuli. So we had to develop all the experimental stuff in order to conduct the experiments. And so once we had all that, this is an example of an experimental protocol for one subject. So we first record the song. We then prepare the stimuli that we are going to be using the day of the experiment. And those stimuli are the birdsong song, our synthesis of the bird song, and then two control stimuli, which are his own song played in reverse, and the song of an adult conspecific, which is a bird of the same species. Then we do a preparatory surgery, and on the day of the experiment, I perform a craniotomy and lower an electrode. We use extracellular electrodes. Right now we are using single units, but are migrating to multi-electrode arrays. And so we then present 20 randomly organized presentations of these stimuli, and well, we then analyze the signals and see what's going on. So we perform these experiments in vivo in anesthetized animals, and what we have are extracellular recordings like this one. This is an example. And we use electrodes that are high impedance. They are from 1 megohm to 3 megohms approximately. And so this allows us to, once we are measuring, to be able to separate in a recording these small spikes, for example, that come from this type of spiking neurons from these other ones that are larger, using a spike sorting algorithm to separate these contributions. And so to give you an idea of the experimental results, here we have the raster plots and the PHTH for one neuron of one bird to the response to the bird's own song. And well, here is the reverse song. We use this stimulus and this one as a control to know that we are measuring one of the HBC neurons that are selected to the bird's own song. So here we see that there is no significant activity when comparing it to this one. And then we have the responses to the synthesis that we perform using the song production model. So what we see here is that some features of the firing during the bird's own song, we are capturing them in our synthesis. The amplitude of the histogram is a little bit lower, so it may be that there are some features that are escaping our model, but we have a good amount of firing. So here are group data for 20 neurons coming from 5 different birds, and what we have is a measure of mean spiking rate across presentation. These are all responses normalized to the response to the bird's own song. So a score of 1 means the same response, and a score of 0 means it didn't respond. And we have a measure of mean spiking rate, mean spiking variance, as well as correlations. And we see in all cases that the synthetic song has a significantly larger response than the control stimuli. So, in order to conclude, and some final closing remarks, our synthetic song is capable of reproducing the replay phenomenon of these neurons when you play back the song to the bird, meaning that the model is capturing some of the features that are relevant of the song for neural coding. And something that I think is important as well is that having a low-dimensional model with clear time-dependent parameters that are related to physiological variables can help us interpret the neural coding of this behavior in a way that, since much of the complexity of the behavior is driven by the dynamics of the periphery, maybe the neural coding underlying this behavior is not so complex as one may think a priori. So thanks again, and this work is funded by CONICET and partially by the NIH. So thank you.
So is there any information encoded in these songs, or do they just...
I'm not here.
That did not work.
Okay.
Is there any information encoded in these songs?
Sorry.
Encoded. Is there information? Is there a message that the bird tries to convey to someone else?
So mainly in songbird species, only the males sing. Mainly, I mean, there are some species where females sing, but so the song is mainly to try and attract the females in the mating season. So the more complex song you generate is the most exotic in order to attract the females. It has something to do with that. I don't know if that's...
You're sure you mean no?
What?
Your short answer is no.
Oh, no. Well, the information is that communication, I mean that, what do you call it? Like sex appeal?
But actually, I think that the experiments where the bird hears its own song, the bird is asleep.
Yeah.
So it's recognizing himself, his own voice. Is an experiment to check that the bird recognizes its
The neural coding that we are showing here is the bird recognizing himself. But the thing is, these neurons fire when he's recognizing his own song in the same way that they fire when he's producing his own song.
Yeah, maybe I'll explain my question. So if you took my voice and you scrambled it somehow and then you played it back, this would correlate with my speech, but will carry zero information, right? So you would measure something which is really not, functionally the correct thing to measure. And so when you show that your reconstructed signal shares correlation with the song, that's fine, but the question is, do you measure really the parameter that is relevant here? Are you reconstructing? Are you inferring the correct parameter?
Well, when you mean correlations, you mean this figure?
Yeah.
This figure right here? So, no, what I'm correlating here is the neural responses. What I'm correlating here is this, which is an histogram of a neural response to this one. So what I'm correlating is neural activity in response to one stimulus and to another. And what I'm showing is that while we're still not at correlation 1, we are, in a way, capturing some of the features of that signal. Yeah.
You want to stay with this? So I just on the dynamic model.
Yeah.
Do you think that's like a canonical model where if you change the parameters, you can get all sorts of bird sounds? A and B. Could you explain intuitively what you were saying about the bifurcation in the limit cycle? Is that a bifurcation that then goes into a limit cycle and then repeats? Is that?
Yeah. So the first question was what?
Well, is this a canonical equation that where you could get other bird sounds by changing the parameters?
Yeah. So the model, where I don't remember where it was.
Yeah.
So by varying this parameter, which is the scaling parameter, we are able to produce sounds from different bird species. We have produced songs from canaries and from some bird species of Latin America. And a recent work has used this same model to produce vocalizations in primates, and similar to primates, by varying only this parameter.
Nice, great. So that's answer 1.
Yeah.
Excellent.
And B.
And then back to the model.
Yeah.
When you just did.
So here is the bifurcation diagram. So region 2 is a region where a stable limit cycle exists in in this system. And region 1, there is no limit cycle, and region 5, there is something in between. But the thing is, if you cross from region 1 to region 2 by this red line right here, you cross a Hopf bifurcation. And so if you do a trajectory that goes like this and then comes back, it's like this. What you have here is varying alpha. Here, P sub is basically alpha. So what you have here is what you will have here is a tonal-like sound because you cross a Hopf bifurcation. And if you cross through here, you get this kind of sounds with spectrally rich content.
I don't know if we have any more time. Maybe one last. Okay.
So I understand that you're able to figure out what the alpha and beta functions are from the song, correct? Have you looked at the just looked at alpha and beta with respect at the same time as the neuronal data, just to see what if there's any obvious correlations?
Yeah. So I don't have alpha and beta trajectories here, but I do have what we reconstruct from the song recording. So alpha and beta, we have at the lab a database that has different alpha and beta alpha and beta values. So you can take these trajectories and reconstruct. Basically, what we look to reconstruct is for alpha in this work, we use only on and off values. Since alpha is related to the pressure, we said the bird is phonating or he is not. And then we modulated this with the envelope to have amplitude modulation of that pressure. And then for the beta, which is related to the tension, and that is another experimental result, by varying the tension, you get higher frequencies. I mean, by increasing tension at the muscles, you get higher frequency in sounds. So what we do is we take the spectrogram and we reconstruct fundamental frequency over time. And so we have a database, and in that database we look for the alpha and beta that can give us phonation and that fundamental frequency at that given time.
Cool. My question was more about, have you looked at the alpha parameters and the beta functions along with the neuronal data, like the neuronal firing data?
No, we haven't done that yet.
Okay. That would be pretty impressive.
I think we have to move on because there is the banquet. So we thank Santiago.